86.
Which way are you going?
Yeah.
74. 7, 6, 7, 66, 62.
I guess that flow's not very good.
Yeah.
All right, gonna
I
come
think
back
you're
down.
well past it now. I think you should go for the closer.
Yeah.
6, 5, 7, 7, 6, 8, 8, 4, 8, 6, 9, 10, 10, 6, 9, 5, 7, 5, 7, 6, 3, 5, 5, 6, 7, 7, 8, 8, 9, 9, 7, 9, 9, 9, 5, 8, 7.
Just keeps going down.
Yeah.
I mean, eventually we're gonna have to take a sample no
coming
matter
closer.
what.
Yep.
So.
Yeah, Amy, just so you know, Julie wrote in to say she agrees with you.
Okay.
5, 5. Yeah, so if we can get up
If
to
we get
about
up to 10, I'm okay. I keep lowering my standards. It's okay.
7, 3.
It'll be different.
Well,
than the
keep
other.
in mind though that the water that we're seeing here, the microbes are probably much warmer, even just 5 or 6 centimeters deep inside that
Yeah.
tube worm
What about here?
bunch. 8, 6, 9, 9, 2, 9, 7, 10, 10, 3, 10, 6, 10, 3, 10, 4, 9.
That's holding pretty steady.
I can go up a
Let's
little
go
bit.
for it.
I wouldn't move just yet.
Yeah,
Roger,
let's go for
holding.
it.
You want to go for it?
Yeah.
Okay.
It's pretty steady. It was. Yeah, it's going back up.
All right, I'm frozen
It's
here.
hovering between 9 and 10.
You ready, Amy?
Yep, I'm
All right.
ready.
I'm taking the sample now, starting temperature 11. Coupling is turning, so the valve is opening. That's a good sign. While it's opening, the temperature is not going to be very good because it's the motor current, for some reason, gives sort of incorrect temperature, so I would not be too concerned about that 4 degrees we just saw. Although this 5 degrees seems to be real.
So for those of you on Nautilus Live, we are currently zoomed in with our Hercules view on an area of diffuse flow coming out of the Seacliff hydrothermal vent sites. For this particular series of measurements, we are just trying to get a stable temperature within a pretty well prescribed range. What increases the challenge, however, is that in these areas of diffuse flow where the vent fluid is mixing with seawater, temperature is measuring over scales of centimeters or less and as a function of time. So that's why we've been spending quite a bit of time carefully kind of exploring the temperature profile here with the tip of this IGT isobaric gas tight sampling system. There's a little thermocouple at the end of that snorkel that measures the temperature, and then we're currently drawing in vent fluid down that metal tube into those pressurized cylinders there. The aim is, at the end of the day, to bring all that material back to the surface at the same pressure it was at depth and study the dissolved gases and minerals contained in therein. For those of you wondering what kind of tube worms and life we're seeing here, inside those long thin tubes, I believe that's Regia pisceci that we're looking at. And then those wonderful flowing gardens of worms you can see just with the cable to the IGT superimposed in front. Those are Alvanella pompehana. the Alvin Pompeii worms.
So, this is Vincent speaking. The IGT temperature seems to make some weird jump in temperature. Is it normal?
Yeah, I was giving it an extra close to be sure it was really closed at 800 milliamps, which is the closing amperage we allow the motor to pull. So sometimes when you do that, the temperature goes a bit haywire. It's a normal thing. Okay, sample taken. You can go ahead and stow that one, please.
Roger. Video, please come wide.
Roger.
So that varied between like
Pull
6
wide.
and 12
Roger.
ish.
Well, yeah,
Degrees?
about that. I'll be able to look when
Okay.
when I go through the text file.
Okay.
I'll be able to give you. I can give you the whole text file.
So we're doing the second one right here. That's correct, right, Sean?
Yes.
All right.
Remember where we parked?
Yeah.
In the
Okay,
bushes.
So just to give you the zoom in on where we actually are in the ocean right now, we're currently 2.7 kilometers below the surface. The sea cliff
Standing over. Yeah.
The sea cliff hydrothermal vents are part of the Gorda Ridge marine feature where the Pacific and Gorda plates meet. The Nautilus itself, 2.7 kilometers above Hercules right now, is itself 180 kilometers off the west coast of North America. It's a bright, beautiful day out there, but inside the control room here it's all dimly lit and everybody is very focused on this task at hand.
Nice. Let her go. Yep. Good.
Looks like she's in on both sides.
You just want to bump it
Roger.
towards us a little bit. Just a tiny little touch.
Not so tiny.
I think it's good.
Yeah. All right, so second IGT.
Yes, please.
Roger. I guess I'll refrain from pulling out the drawer a bit there.
So our Hercules pilot Jessica is controlling that craft manipulator arm using a scale model that sits on the desk here in the control room, and every movement she
Video, can you
executes,
push in a bit there, please?
Roger.
every movement she
That's
executes
good, thank you.
with that model is replicated in the arm on Hercules. So what we'll be doing next is actually
Nice.
just getting a second sample with a second IGT at that exact
Alright.
same site.
Video, you want to come wide again there, please?
Mm-hmm.
That's good, thank you.
Pull wide.
Thank you.
It's good experimental practice to always make sure you've got a backup sample. It's not easy to come back a second time on a different visit. And I guess you were telling me, Sean, that saved you many a time, taking that second IGT.
It saved me on this cruise.
Oh, really?
Yep. IGT-7 on the way down on the last high temp dive. The valve didn't seat correctly and it filled with seawater on the way down.
Oh dear.
Just move your mic a little bit, Sean.
Could you not hear me?
You're pretty faint.
No,
You're pretty
I think
faint.
sometimes
Rotate
it's the
some
angle
more.
of the mic too, because I see it's very close to your mouth, but
So is that
Yeah,
too
there.
close then?
I can't.
Better,
No.
worse?
Try one more time.
Can you hear me now?
No.
I think sometimes when you move, like the cord might move. Maybe it's like the end of the cord or
There's a
something.
connection. or
Yeah.
something.
What do you want to do? Look
Is
up
this
more?
any better?
No, actually, it's
Very
alright.
odd
Pretty
that
good
it's so intermittent.
I didn't
Yeah.
actually
Yeah,
move it.
sometimes
No,
it's
I
very
know.
clear, and then like right now, I'm only hearing you because you're close to me.
Right. If I were any closer,
I
I'd be
know.
eating
No,
it.
yeah, it's not. It's
Yeah.
not you.
Not you.
it's the
It's
mic.
the mic.
It's not you. It's the mic.
It's not
Alright,
me,
video,
it's
you want to
It's
come
not
in
you,
a
it's
little bit
me.
there,
Yeah.
please?
What you could try, Sean, if you have a chance, is to mute your mic and then just you could feel the mic and see and rotate it so that I think the flat part needs to be facing your mouth.
How's this? No? I have the flat part. Actually,
Sean is now eating his mic.
Isn't there 3 headsets over there?
Yeah.
Yeah.
Seems, Sean, you always get unlucky with the headsets over there.
Yeah.
What does that read off as? Can't really see the snorkel very well.
13.6?
You can see it bubble.
Or is that the last one?
That might be the last one.
You can look at bubble there. I'll give you some zoom.
Yeah.
You ready?
Video and a
all right, how about now?
Yay!
Welcome back.
11.5.
Probably had never had
It's good
so
to be
many
back.
people be happy to hear your
We
voice.
missed you.
I know, that's not usually the case. Especially my kids. 11.8. It's a pretty steady 11.6 right now.
Ooh, tempting.
Are you in a good spot, Jess?
I'm frozen right now, and it's like the
What do you
load
think,
is
boss?
good.
Sure.
All right.
Wow.
I like it when it works this way.
That was fast. Yeah. Knock on wood.
It's a very solid 11.7.
Roger.
Cool.
So it's starting the sample now. RGT1, yep, okay. Coupling is moving, good sign.
Hey, Sean, are you able to rotate your screen just slightly this way? I was able to see it before just because I'm keeping track too. Thanks.
So Sean, that rotating coupling, that's opening a valve that allows the seawater to then be drawn into the chamber?
That's correct, and it's drawn in just based on bottom pressure alone. There's no physical or mechanical suction that allows it to go in.
Okay, so you've got 270 atmospheres of pressure on the outside and 1 on the inside.
Correct.
Okay.
Well, not quite one, but
Around that, yeah.
Nicole, is there a screen back there you'd like me to put Sean's temperature readings on?
Oh, you know, it's okay. I can see it from his screen. Thank you for asking, though.
Okay, yeah.
Hey, Alex, how's the ship doing?
We're good. 18 knots of wind. It's holding steady.
Roger.
Sarah, if you have a moment, we do have a question about the right monitor in Zaur's seat, so science seat.
Yeah?
It's just not displaying anything.
Okay.
So for those of you on Nautilus Live, we're just taking a second vent fluid sample at this diffuse flow site.
Okay, if I move the wall real quick.
Sure, go ahead.
To give you a sense of how zoomed in we are, those two laser lights you can see on the Hercules view, they are 10 centimeters apart. So your total field of view here is about 50 centimeters by, let's say, 35.
Sure.
It gives a great visual impression of the abundance and diversity of life that can be hosted in such small regions by these diffuse flow sites.
Okay, Jess, you can stow it. Great sample.
Awesome.
Good solid temperature the whole way through it, looked like.
Roger.
Does
Yeah.
drop a little bit during sampling, but that's normal.
Okay, sounds great. Are you going to come wide, please? Thank you.
No problem.
Oh!
It's like, oh, that's magic.
Wow!
Oop.
Josh, so many hats.
I thought
Yeah.
Sarah had perfected
More
mind
controls.
control.
Give me more buttons. I want more.
All right, so it's super time.
We might need to pop off of here and then
Yeah.
jump back down to get the super wand.
So that means maybe once we're set up, Sarah, if we could put bubble cam on the wheel and then we'll send that to shore, but it could wait until they're all situated.
Okay. Roger.
So Josh, did you guys cut grooves into that crate so that these guys slot in?
Yep, that's right. This little homes for the wand end and the body of the IGT to kind of nestle into.
Yeah, that's pretty cool.
Yes,
There you go. Done.
it's okay. Nicole, what's our depth?
Depth.
2707.
Yep.
Thanks.
If it's in there, it's good.
Our bubble's now on the sat feed 3.
Roger.
Thank you.
Just gonna halt the harm here. Halt the harm. I'm gonna halt the arm here and then we'll fly up and grab the wand.
Lounge van.
I want to pull it out for you while you're hovering.
Sure. Sounds good.
Am I quiet again?
Sorry.
I can hear you.
No, we can hear you. You're still with us.
I was just looking for Sarah if she was in the lounge.
Are you
I'm
also
here.
on SBL? Okay.
Should be.
Yeah, she heard you.
Oh,
Hey, Sean. Sarah's here in the lounge. Hi.
hi. Can you come up to the van?
So Nicole, and just to confirm, I can come off and we can pick up the wand. Do you guys don't need anything else here right now?
No, as long as we can land right here, that's our goal is to get the super in the same spot. So
Roger.
Sarah's on her way up.
Thank you.
So just for those of you following us on Nautilus Live, you'll hear us reading off a lot of values of temperature and depth. Those are all in
Try my auto depth again.
SI units. So that means that the depths are all in meters and the temperatures are all in, well, degrees Celsius.
Sorry.
If you get a chance, would I be able to reset DVL?
You want to hold off on that for a minute? I'm
Okay.
just in auto XY for a sec.
No problem.
But when we're setting down, then you can definitely...
Got it.
So we're going to be staying at this vent for the next little while. What we're doing now is just extracting, well, we call it the wand. It's a combination of an input hose for what we call the super sampler.
Come up a little bit.
Roger. Coming up. Sorry about that.
Right.
It also has a temperature probe on the end, so we can register the temperature of the fluids that we're sampling. Just to get it out of its holster, we need to get off the sea floor just to extract it from its storage place, and then fly back down to the diffuse flow site we were looking at.
If you want, we can just hold it in our arms while we sit down.
I'm just going to make sure I got a good angle for downward. Yeah, that should be good. Okay.
All clear for me to go sit down?
Yep.
Roger.
So just a quick update for those of you tuning in on Nautilus Live. We are currently at the Seacliff hydrothermal vent site. Today's focus is on sampling vent fluids and collecting a few geological specimens. We're currently measuring vent fluids using a couple of devices at this diffuse flow site. The Seacliff hydrothermal vent site itself is part of Gorda Ridge, which is a tectonic plate boundary 180 kilometers off the west coast of North America. And we're currently operating at a depth of pretty much 2,700 meters.
Let's come back this way a little bit.
Yeah, I'd rotate a little bit to starboard. Yeah.
Free up the arm.
Yep.
Yeah.
Yep.
Our ROV pilots are just making sure that they return Hercules to pretty much the same spot it was at when it took the previous vent fluid samples with the isobaric gas tight sampler. And now we're going to sample the water again.
See how it steadies out real quick though, Josh.
Sure. I'm just getting the wand out of the bushes there.
Sure thing.
Got a tube worm right through that loop.
Yeah, maybe you want to come up a little higher.
Yeah.
Yeah.
Let's try right here.
Yeah, keep sliding over a little bit.
Sure.
Yeah, you can come up a little. I kind of put the porch on top of that sort of little.
On top of this little clumpus?
Yeah, because we'll sink down again.
Roger. Oh, it might be right where we want to sample though.
I think we're in. Yeah, here. Yeah, come down there. We're right above that little bunch of worms there.
Yeah.
Yeah.
Okay. See how this pans out. All right, you're in stick lock currently.
Okay.
So this device is what we call the super sampler.
Pan up.
Roger.
See the arm.
And no matter how many times...
I go rock back all the way.
Roger.
And then just go to the right. And then look up.
And no matter how many times the science team in the back row tell me what the acronym stands for, SUPR, I seem to be unable to remember it.
It's Suspended Particulate Rosette Sampler.
Thank you very much.
Got those acronyms down over here.
Yeah, data.
So we're going to be pumping this water through a filter, is that right? And collecting
all the way up to the bumper bar?
That's
Roger.
correct, John.
Yeah, collecting hopefully bacterial, well, microbial samples from the vent fluid.
Come down a little bit.
Last time we caught a limpet.
So I'm gonna go this way, this way.
There you go. I'm gonna look down now for you.
Sure.
So I wasn't in the van when we first arrived at this site. Was there much discussion of the fact that these tube worms are kind of some of the longest we've seen in this particular location on our dives?
Not really.
Okay.
I don't remember talking about that.
No, I was just curious. It's just striking to me that so these are some of the longest and thinnest tube worms we've seen at the Seacliff site over the last few days.
Go ahead, Data. Stand by.
I think she
Okay.
said stand by.
I think it's pretty close to where we were.
Yeah.
Might have to just move around a little bit.
Roger. You want to push in on that one a little bit there, video? That's
Yeah.
okay with you, Josh?
Yeah. Oh, losing
That's
my
good.
Argus view here.
Yeah, I can
You
fix
get
that.
I can
for
zoom
you
out on
live.
Argus a bit.
Yeah, maybe.
Yeah,
Just gonna get it fixed up.
Data Lounge.
Getting a max of 5 on the super temp.
Okay.
And about 5 or 6 on the thermocouple.
We're
7,
just going down to find Ashley, and we'll send her up to you.
8.
Just gonna try and straighten it out a little bit here.
Okay. Just try not to suck up too many limpets.
Right.
Might stop the flow meter.
Is now an okay time to look at the flow meter, Josh and Jess? Or do you want to keep?
Let's grab a temperature first, and
Yeah.
then we'll flip it over.
We're around 6.
And this is the super temperature that's on the super, not the additional redundant one.
They're both showing around the same.
Okay,
Yeah.
got it.
The one with the orange panel is the thermocouple from the
IGT.
Yeah.
Okay, sure.
Still 6.
Okay.
Seven 7.6, 8, 9, 9.7, 10. Looks like a good spot.
Okay.
10.8,
Not
11.
too deep in the weeds there.
Those look mostly like worms. 12.8,
We're at 12,
12.9, 13.
but not the
So
super.
maybe we can just pull back just like a
Just a smidgen.
centimeter.
Sure. Yep. Okay, centimeter. I'm gonna move the.
This is at 14 now, flow meter.
Yeah. We're at 13. $12. Can you tell if we're in limpets?
I think we're clear of them. I think
Okay.
so. Yeah. Just I came up that tiny little bit. And
Okay.
then that dropped the
Awesome.
temperature, but I think I'm still.
Okay, so we're holding steady. The thermocouple says about 12.6 to 13, and then the super temps at 9.7 to 10.
Is that what you saw previously? The difference between the two?
This one, the super one, is a little bit slower. It's not as
Okay.
quick to respond. Let me get my timer set.
Come down a little bit on Argus.
Roger.
Okay, so we're gonna filter for 20 minutes. We're at 12.5 on the thermocouple. 10.4 on super temp. Okay, I'm going home.
Can we just tilt the camera up so I can see just the bit of the arm?
Sure. You want to come a little wide there, video?
Yeah.
There you That's
yeah.
good.
Maybe down just a wee bit. Just want to keep an eye on the wrist because I'm in continuous mode right now.
Yeah, that's fair.
A little wider?
No, that's perfect
No,
right
that's
there.
good.
Great,
Okay.
thanks.
Okay, I'm at port position 1.
Temperature's still steady.
And start filter. Filter
Start.
slow.
Whirly gig moving.
So on Nautilus
It's
Live, on the,
tricky.
Yeah.
Wand is tricky to get in the right spot.
Oftentimes it kind of
Yeah.
slows
Get
down
up,
at first
up,
and then it'll pick back up, so we'll
Yeah.
have to watch it.
It's elastic.
So you got to be able to move your elbow down. You got to pitch up and then yaw over and then get the wrist twist in the right position so you can get the elbow down.
It's moving
Yeah.
pretty slow.
So
Yeah,
you
it's
can
like
make up
longer.
for your elbow by 3 different joint movements.
It's true.
so on the Nautilus Live quad view, you can actually see the
Come on, flow
pumping
meter.
mechanism operating. Are you assisting it with the power of your mind, Amy?
Yes, I am.
Okay, good. Those of you on Nautilus Live, feel free to join in.
Video, you can push it on Argus a little bit.
All right. There you go.
Right there, yeah, that's groovy.
It's a cool shot.
I'm gonna turn off one of the lights too and see.
Nicole, can you see if Eric's in the lounge? If he can.
You can really see how big the tube worms are compared to Herc. They're pretty tall.
You're doing pretty well, Amy. Your concentration's keeping it moving.
Thanks.
Yeah.
Temperatures 10.2 on thermocouple, 8.18 on the super.
Amy, do you want me to go try to find him?
Oh. Maybe.
Yeah? Okay.
Well, it seems like it's, I don't know, I can't tell.
It's spinning, just slowly spinning. but
Yeah. Oh, okay.
It's like
Did
I'm
you
just
say
not sure if stopping
it
and
will
starting
come a little
again will
wider on
help,
Argus.
or-
It's not so like.
Did
It
you say
seems to be going
10.3?
faster now. 0.7. It was 0.4. Sorry.
Sarah, did you copy that?
It's
It'll
okay.
just come
never
a
mind.
little wider on Argus. We don't
Yeah.
get so much of the ship movement there. Yeah.
This is for filter 1.
Yes.
So each filter is 20 minutes, you say?
Yes, we've bumped it up from 15 minutes
Thermocouple is at
to 20.
15C.
So for those of you watching on Nautilus Live, this is where we don't have a chance to get to all of your questions and comments that you're posting to us, but we'll try to feed them in occasionally. But at times like this, we let operations and science take the lead.
The holding. Oh, we did this for hours last time.
It's true.
So Josh, when you're sat here for 20 minutes or so, do you kind of have a freeze mode on the arm?
Yeah, so we can, you can halt the arm, which is I'm in a halt mode now. So you can basically hit a button and that will lock all the joints and hold them in that position.
Temperature's at 17 C now.
And then you just kind of monitor
Which
it.
probe?
So I'm kind of ready.
The thermocouple.
Ready to come live with the arm again in case I need to do some small movements.
Now it's at 18.
So I still kind of hold it in the ready position, but I'm relaxed, like my hand is relaxed now.
Okay.
And so I have a grip lock on.
What is that?
This is when we
and
started.
halted in that position.
Okay, because my...
This is when you started.
Do you both have side chat over there? Okay, yeah. Julie's just saying it looks really steady and looks good.
Thanks.
Yeah.
I'll keep this open.
Yep. Come up just a tad.
Roger.
Okay. It's fine.
So at the moment, for those of you watching on Nautilus Live, we are currently zoomed in on a site of diffuse flow at the Seacliff hydrothermal vent site. We are using this wand-like apparatus being held by the Hercules manipulator arm to essentially pump vent fluid through a system we call the super sampler mounted on Hercules. That vent fluid is being passed through a filter, and we're hoping via that filtering process to capture any microbes that are entrained in the vent fluid and possibly coming from sites deeper in this diffuse flow site that we can't actually physically get to.
Nicole, it's lounge.
Go ahead, Darlene.
Eric's on his way up. Over.
Thanks.
We'll be here for about another 15 minutes or so with this particular sample.
Okay.
To give you kind of the zoom out of where we are and how we're exploring, Hercules is currently 2.7 kilometers under the ocean at this Seacliff hydrothermal vent site. This is one feature that's part of the wider Gorda Ridge area that is where the Pacific and the Gorda plates meet, approximately 180 kilometers off the west coast of North America, just where California meets Oregon. And I know some of you have been asking, when we talk about the control van that we're operating in, this is a structure on the top of the Nautilus.
Data. The super's at 15.
The temp?
Uh-huh.
Yeah, okay, got it.
And thermocouple's 13.5.
Okay.
Alex, can we do a 5 meter move 270?
And so in that control van, there's approximately 10 of us, each with a specific
Bridge
role.
nav.
ROV pilots, navigators, video engineers, and scientists.
You get 5 meters at bearing 270.
So if it's a convenient moment, I was just going to pass a quick question to the back row. What are we going to do with these filters once the ROVs return to the surface at the end of the day?
Once the filters are on board, we actually use a solution called RNA later. Put those filters in a tube with this RNA later. And then we do just that. We extract RNA later in the lab.
Okay.
Once we have that RNA, we can do a lot of sequencing work and bioinformatically get an insight into what microbes are present in this environment and what metabolisms they might be involved in.
Uh-huh. Now, is that what you will actually do in your mini lab on board, or does that wait until you get back to Woods Hole?
That waits until we get back to Woods Hole. Because we need a pretty, yeah.
That's cool, thank you. If you're watching on Nautilus Live, channel 2, or the quad view, top right, you get a great view of how Argus is gently moving up and down in the swell as it's tugged at the end of the cable deployed from the Nautilus. But then you also get a great view of the 50-meter neutrally buoyant tether, that yellow cable that goes from Argus to Hercules. That just goes up and down in the water column as Argus goes up and down, leaving Hercules completely undisturbed. So Hercules is basically now parked right in front of this site of diffuse flow, where you can see all these tube webs in front of us, and is able to operate as an entirely stable science platform on the seafloor. So Sarah, your answer there provoked a great question on Nautilus Live, and I was just going to throw it out to you quickly. Why do you sample RNA and not DNA?
We do actually sometimes extract both the DNA and RNA. Maybe Amy can chime in too on when she's done RNA and DNA sequencing work. But the RNA is going to give us a better idea of what microbes are active, right? Because the DNA signature, we can still get metagenomic information from the DNA. Gives us an idea of you get the genome, get metagenome assembled genomes from the DNA component, but the RNA will tell us what genes might be expressed in an environment, what microbes are, if they're expressing a gene, then that gives us a better idea of what metabolisms they're actively involved in right now when we're sampling.
Okay, that's great, thank you. I think Amy's still using telekinesis to operate the super, so she's fully occupied.
I'm conferring with Eric too.
I'm sorry, just ignore me.
It's okay. We're just making sure everything's running smoothly. So we still have 8 minutes left for this filter. No, it's air, probably. I haven't seen anything go through the wheel. I don't know.
Did you see it in the flow? Is that what you were saying?
Yeah, I think it's okay. Just sometimes there's a little bit of air in the line, and then once it's trying to pull this fluid through a 0.2 micron filter, and which are really tiny holes, and so it kind of slows down, and it's really hard to pull all that fluid through. So when we fill up the bags, the wheel should move a lot faster, about 3 times faster or so, because you don't have to pull it through that filter. And then if you start accumulating biomass or suck up a tube worm or something like that, that's also going to kind of get in the way and slow everything down. And we have like we want to get as much biomass on the filter as we can, so it's not difficult to get what we need out of our assays. So So we try to go as long as we can without being here for 2 hours. Yeah, I agree. We're running 20 minutes per filter. There's 6 more minutes on this one. Okay. Okay. So Eric was saying after this filter, we're going to try to push it and make sure it's going in the right direction and make sure there's nothing stuck in there. So we can kind of push it out. When I flushed this morning, I did see lots of air bubbles coming out.
That just popped up.
We're at 14.9 for the thermocouple and 13.2 for the super temp. Yeah.
No.
Can we see where the end of the wand is? I can't really see it anymore.
I could show it to you in bubble, but then you're going to lose your twirly thing. We could try and zoom in down there if you want.
Okay.
Have a look at it.
Yeah,
sure.
that'd be great.
Bit.
Yep. Go for it. Get a good
push
zoom.
it more. This is full zoom.
Yeah, I think it's okay. Look at all those beautiful things.
Yeah, there's a great view there of the Albanella tube worms.
You can see it's kind of, well, it's hard to tell, but I think it's just above the surface of the worms.
Okay. Yeah, I see the tip of it there,
yeah.
The dark blue.
Is that all right there? Can we go back to where we were?
Yeah.
Okay.
Thanks.
Okay, zooming out.
Step out a little
wide,
more.
I think.
Tilt up. That's
So
good.
Flow rate's constant. Oh, it's up a little bit.
So for those of you watching on Nautilus Live, on the quad view in the top left, you can actually see the zoomed in view of Hercules' manipulator arm holding this super sampler wand that's at the moment pumping water from the vent fluid, pumping vent fluid through to the side of Hercules, where it's going through a series of, I think I heard you say, Amy, is it 0.2 micron filters?
Yes, that's correct.
Yeah. You can actually see that pumping mechanism in the quad view bottom left. You can see that little wheel turning that indicates the flow direction and rate. And then in the top right view, you can see the view from Argus as it looks down on Hercules.
Thermocouple is at 15.8. Super is at 12.1. Flow is looking good. And we have 2 minutes left on this filter.
What does the filter actually look like when you get it out of the super? Can you see kind of discoloration on it?
So the first time we sampled, we did see one of the filters kind of had some black precipitates on it.
Uh-huh.
The others were not quite as darkened. You want to be able to see something accumulating on it.
Yeah.
Because that's a general indication that you've got some biomass and you can get lots of DNA or RNA out of it.
Yep.
And then one of the filters had a limpet on it as well.
Oh.
So
Sorry.
I had to take that one off. But,
Uh-huh.
I'll kind of interfere with our measurements.
Wait,
Yep.
okay. I was going to say, I don't see, yeah, on one side of the filter, just before he went through the 0.2 micron hole.
Right.
Yeah.
Yeah.
We got about 45 seconds left.
We've got some eagle-eyed viewers on Nautilus Live who've noticed that since we've left the tube worms alone over this 20-minute period, they've gone pink again, and they've actually come out of their tubes at the end.
Yeah.
Oh, that's a nice observation.
Yeah.
It's kind of like when you walk through a forest, all the birds stop singing, but if you stand there long enough, they'll start singing again.
Yep.
Mm-hmm. Does that mean tube worms are the birds of the hydrothermal vent land?
Exactly. I bet you if we listen hard enough, they will sing. That's not real science, I'm sorry for those.
We'll see if we pick it up on the sonar.
Exactly.
Okay, 20 minutes is up. I'm stopping this filter.
Thermocouple temperature is 11.5.
Let me put down the flow counts real quick.
Looks like the maximum temperature we reached was 18.1.
You want to... Josh, can you pull the wand back so we can see it?
Roger.
Yeah. We're going to try to blow it out.
Got it. You want to come a little wide there on Herc? So you can see the arm. We need to zoom in on the wand.
Sure.
Oh, never mind.
Do you want to come wider or zoom in?
Zoom in on the tip of the wand.
Okay.
Okay, move to 1.5.
That's fine.
Do we need to pull the wand out of the shimmery fluid? So we can see it? Oh.
That's good right there.
Okay.
It's like an anemone.
Yeah. To give you a sense of the scale, the tip of that wand is about 1.5 centimeters across, maybe 2 centimeters.
There's not.
Is there any sediment that you could point it at that would blow away if we blew at it?
I don't know about sediment, but there's like
Or-
some worms there.
How about a tip of a tube worm?
Something
Okay.
that would
Should be
move.
fine.
I mean, it looks like the wheel's spinning pretty fast.
Oh yeah,
Yeah.
that's going for it.
Yeah.
Oh, there,
yeah.
yeah.
Blue water on it.
That worked.
Okay.
The tube worms are moving.
You won.
Yep.
Yeah, blue water
Okay.
on him too. Yeah, got some flow.
It's a good game of whack-a-mole.
Yeah, it's definitely a whack-a-mole game. Yeah.
So just a quick shout out to,
Find that spot again.
I think it's Middle College High School. I see you're getting loads of questions on Nautilus Live. We'll do our
Yeah.
best to get
Yeah.
to them.
There's
Yeah.
the mole. He's coming out again.
Okay.
Get him.
Okay. Okay, so let's go back to the spot, the sampling spot, and then we'll try to move to filter 2.
Okay, that could become a little wider, maybe about a half, a little more. That's good there. See if I can do this without bubble.
Okay.
zoom in just a little bit on the tip.
Yeah.
Just tilt down a little.
Roger.
Just let me know if you get a temperature that looks good.
It doesn't seem to be moving.
Oh, I'm sorry.
That's
Here.
okay.
All right, now it should be logging the temperature.
Okay,
I just switch
thermocouple
over.
says 11.16. It's going up. 13.9, 14.6, 15,
Okay, let's come a little wider again.
Roger.
14.6.
Tilt up a little. It's that same thing where I can see both. Maybe pan over to the right.
Roger.
Up a touch.
What's this one?
Not too far.
Maybe
Okay.
down a touch. A little more bump. Just come a tiny bit wider.
All right.
Good. And then we'll bump up one notch. Actually, that's pretty good right there.
Roger.
I'm in slave now, so I'll see if that's... It was creeping just a tiniest bit.
Yeah.
Thermocouple is at
Put it back into slave
18.3,
mode, so.
which was the max for the previous sample.
This is nice
now
here on
19.
the wrist because then you can
Ooh.
see if you're in the middle of the pot.
Oh, nice.
Right?
Yeah.
So then you know if you're all the way over to one end, you have to flip it over
Looks
to
Yeah.
bring
pretty
it back to
steady.
the middle. So I knew I was in the middle, so I could go to continuous without
Yeah,
being
I
on
agree.
one side.
I see.
Now it's at 20.
20. Ooh, hit the hot spot. We've been looking for that spot for a while.
Yeah. Found it. Okay, it looks pretty steady around 19 and 20 for the thermocouple.
Oh, sorry.
I'm sorry.
The super is at about 14. Okay, I'm gonna advance. Are you in a good spot?
I am stable, locked,
Okay.
ready.
Okay, I'm gonna advance to port 2. And I'm going to start filtering slow now.
So we're now collecting a second sample of vent fluid through the super sampler. If you're watching on the...
Here, why don't I hand you this and then I'll do some numbers.
Roger.
Oh, except I don't have the bubble, but whatever.
Yeah.
Huh?
Get everything I can.
Roger.
Thermocouple temperature is at 18.7, now 19.
So for those of you watching on Nautilus Live, as we're conducting the second filtering experiment, let me give you a quick sense of where we are and what we're doing. So what you can see in the Hercules main camera view is a zoomed in view of a dense growth of tube worms around the site of diffuse hydrothermal vent flow at the Sea Cliff hydrothermal vent site. The manipulator arm you can see is holding what we call the super sampler wand, which is basically an inlet tube for vent fluid. If you look on the quad view, bottom left, you can see a little whirligig slowly moving round. That's the vent water that's basically being pumped through a filter system. That filter is going to be collected when the ROV returns to the surface and studied for any microbial
It started at 0.4 and then it went up to 0.7.
samples that we've collected. This whole site, which we're exploring, is 2.7 kilometers beneath the surface of the ocean.
This is it, total flow counts.
Geologically speaking, this is where two tectonic plates meet, the Gorda plate and the Pacific plate.
Amy, while you're talking about flow counts, when you get a second, can you tell me the flow rate and final volume from the previous sample?
Oh, sorry.
No worries. When you get a second, if you're busy.
Is that?
Yeah, 1 second.
I was trying to be really gentle with that.
So this Gorda Ridge feature is approximately 180 kilometers off the west coast of North America. That's the region of the eastern Pacific Ocean in which the Nautilus is operating. Just to remind some of our viewers, the Hercules and Ares ROVs
I think so.
are remotely operated vehicles. They sit at the end of a long cable that stretches for 2.7 kilometers off the stern of the Nautilus down to Argus and then to Hercules. That transmits